Hi class, this is Miss Durley. This is part 1 of Unit D, DNA. Some key ideas that we're going to examine in this screencast is to try to strengthen our understanding of how DNA's code provides the blueprint, the instructions for building all life. And the other amazing thing about DNA is that this code is going to be universal. So we can take DNA from a human cell and we can add it to the DNA of a bacterial cell, and that cell speaks the same language. So that cell will be able to build a protein even though it is a gene coming from the human genome, which is pretty amazing. So let's get started. Some key vocabulary words. Nucleotide, we recall from Unit C, the nucleotide is the subunit, the building block, the monomer for the polymer of DNA and RNA. Complementary base pairing is the base pairing rule with which the two strands of DNA come together. We recall this base pairing rule is A will always be base paired with T, and C will always be base paired with G. And maybe we don't recall that, but hopefully we will soon enough. Hydrogen bonding are going to be the bonds that keep these two strands of DNA together. A chromosome and a gene. The chromosome is the structure that we see within the cell when the cell is dividing, and a gene is the meaningful sequence of the DNA bases found within the chromosome. So what are the functions of DNA? What do they do for us anyways? So what does it do for our life? Well, it codes for all the proteins. So we remember back to our last unit when we talked about the primary structure of a protein. The primary structure, the sequence of those amino acids, is coded for by DNA's bases. So the bases that are written in your DNA tells your cells how to build your proteins. As well, DNA, one of its functions is that it replicates, it makes more cells. This allows for repair of damaged tissues, growth when you started as one cell to grow into your trillions of cells that you are today. It also mutates, and in our minds, mutation is a bad word, but mutation allows for variation and allows for change over time. So this key idea of biology, all of biology, central that the genetic code is what codes for, so the genetic code is written in DNA bases, and the DNA's bases are A, C, T, and G. They are going to code for this sequence of amino acids, which we call the primary structure of the protein. And if we recall from the last screencast, the primary structure of a protein is what determines that protein's function. So some terminology, and sometimes this is tricky for some people. So we want to be sure that we understand that a chromosome is a structure of DNA, and DNA then is the material. So when we see a chromosome, it's telling us that the cell is dividing. When the cell is not dividing, we call it chromatin, and it's going to appear thread-like, like a bowl of spaghetti. So chromosome or chromatin is the structure, and DNA is the material. Like I can have a t-shirt. T-shirt is the structure, is the type of clothing. My t-shirt can be made of cotton, it could be made of polyester, synthetics. That would be the material. And within the chromosome or within the chromatin, we have meaningful sequences of DNA, and these are called genes. So genes are going to be the sequences that code for one protein. So again, chromosome, the structure, DNA, the material, gene, a sequence of that chromosome. So the human genome is quite large, right? 3 billion bases, 46 chromosomes, 23 we got from our mom, 23 we got from our dad. They united to form a zygote, and together we have 46, 23 pairs. So each chromosome has a homologue, a matching chromosome that codes for the same traits, codes for the same proteins, but one is a version that we got from our mother and one is a version we got from our dad. So DNA is a polymer of nucleotides. We want to recall what a nucleotide looks like. So we remember in the case of DNA, it's going to be the sugar deoxyribose, and that's where its name is coming from. It's going to have a phosphate, usually drawn as a circle, and then a nitrogen-containing base, and we're going to look at those bases in just a second. So nucleotide, nucleic acid, we want to have those words clear in our mind. So DNA's nucleotides, they vary in the base. So everything else is exactly the same. The sugar, deoxyribose, and there's that phosphate group. Adenine, thymine, guanine, and cytosine are the 4 bases that allow for the code, for DNA's code, that writes your genetic information. So 2 types of bases. A single ringed base is given the word pie. I emphasize the pie, pyrimidine. It's a single ring like pumpkin pie, apple pie. Pie is a single ring, right? A purine, thinking of pure, is 2 rings. And if you think of somebody getting married, it's very pure. You both wear rings, right? And so if you need help to remember those 2 words. Putting it all together to build our DNA ladder. It is a double helix, and we want to understand that it has, just like a ladder does, it has rungs. The rungs are built in the middle of these complementary bases held together by hydrogen bonds. So in the rungs, so just like in our ladder, if we were going to climb up a ladder, in a DNA molecule, we're seeing 2 bases, and in the middle, hydrogen bonds. Also, like a ladder, it has a backbone on either side, and the backbone is referred to as the sugar phosphate because it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down the backbone of the molecule. And this molecule, because it is a double helix, has 2 backbones. However, the 2 strands of DNA run in the opposite direction to one another. You can see in this case, the sugar is running this way, and in this one, this strand, the sugar is running this way. So we call that anti-parallel. So these are the parts of the molecule, the rungs. These are the bases held together by H bonds. The backbone of the molecule is formed between sugar and phosphates. This forms the backbone. So the ladder is a nice image for us to remember. So our next screencast, we're going to look at DNA replication, how DNA makes copies of itself to allow for cells to divide and have the same information inside of them. So from this screencast, hopefully you can label a molecule of DNA, the rungs, the backbone. You can explain some functions of DNA, and in general, you could describe its structure. It's made of nucleotides, and you can understand those words DNA to chromosome to gene.